Good afternoon, everybody. Welcome to the Leukemia and Lymphoma Society's webcast called Immunotherapy is Using the Immune System to Tackle Cancer, a Reality. This presentation is part of a series featuring the latest advances in blood cancer research. Let's start the presentation. My name is Andy Fertino, Regional Director, Quebec Division for the Leukemia and Lymphoma Society of Canada. I will be the host of this online event. During the webcast, you'll hear only my voice and the speaker's voice. Since there are many of you online, questions may only be asked at the end by using the online chat function. The presentation will last 45 to 50 minutes and will be followed by a 15 to 20-minute question period. The presentation is being recorded, and you will be able to listen to it again on our website while following along the PowerPoint presentation. We will be sending you an email once the presentation is available online. Our mission is to cure blood cancers and improve the quality of life for patients and their families. We offer support to people affected by blood cancer every step of the way. Please do not hesitate to reach out to us if need be. We have several information tools on our website. You can visit us at lscanada.org. The Leukemia and Lymphoma Society of Canada is currently financing 41 research projects in 2017-2018 and has invested a total of $36 million in research since 1955. More than 138,000 Canadians live with or are in remission from a blood cancer. I would now like to introduce our speaker, Dr. Denis-Claude Roy, who has generously accepted to share his knowledge with us. Dr. Roy is Professor of Medicine at Université de Montréal, Director of Research at CIUS de l'Île de Montréal. He is also CEO of CellCan Regenerative Medicine and Cell Therapy Network, as well as Scientific Director of the Center of Excellence in Cell Therapy. Over to you, Dr. Roy.
Hello, everyone. Thank you very much for the very kind introduction. We are going to talk today about immunotherapy and its use to tackle cancer. And that is really one of the, I would say, the largest breakthrough that we've had recently in the area of cancer. This is really a predominant factor in the area of leukemia and lymphoma, but you also have to understand that this is also an area of major development for the whole oncology field. And while we're used to having drugs to fight cancer, we're going to talk about a different strategy. And you know that drugs are quite effective at cancer, but still large number of patients either fail therapy or relapse, and therefore there is a need for developing novel, innovative, and effective treatments for all patients. One question that's being asked is the immune system going to replace standard chemotherapy? And at the time being, what we are saying is this is used sometimes in replacement, but most of the time in conjunction with chemotherapy, before or after. And the idea is really to use all of our armamentarium in order to fight this deadly opponent. What we will be talking today is the use of cell therapy for anti-cancer, and there's nothing like an image. And here you can see immune cells from your blood, the peripheral blood, the blood that circulates in your veins, goes throughout your body. There are some of these cells that we are calling white cells. Here they're kind of orangey, but they are really what we call lymphocytes, so small white blood cells. And here they are attacking, in blue, a cancer cell. So not only are they on the surface of that cell, but they are actually transmitting factors that are yielding the mortality of this deadly invader. Now, most of the advances that we have done actually stem from the use of a strategy that we've been incorporating in our treatments over the last 50 years. And this is allogeneic hematopoietic cell transplantation. This is one of the most important medical breakthroughs of the last 50 years. And it can cure, as you can see here in this slide, approximately half of patients with refractory blood cancers. And by far, it's one of the most effective form of cancer immunotherapy. And it is very interesting because we are starting to see how it works. And the stem cell transplantation is curing patients through a mechanism which is really immune. And more we are finding that a large proportion of beneficial effect of this strategy is actually the capacity of donor cells to react towards antigens, some of which are called minors, the compatibility antigens, and we will refer to them in this talk as MIAs, or other antigens that are actually present on the surface of cancer cell and are being recognized by the immune cells. Now, we're going to talk a little bit today on how we can take advantage of this activity to fight cancer. So before we start, let's look at what happens with stem cell transplantation. So here you have a patient with cancer. So cancer is schematized in yellow, so you have the cancer cells here. And you have, for example, a patient with leukemia that had treatment and relapsed, or, for example, a patient who had initially leukemia, but we find that although it is responding to chemotherapy, the chances of being cured with chemotherapy are very low, and therefore we'd like to have a very intensive treatment in order to be able to cure, not only to control the disease, but actually to cure the patient. Now, what happens with stem cell transplant is that we are giving high doses of chemotherapy with or without radiation therapy in order to eliminate those cancer cells. But when we're doing this strategy, we are not only eliminating the cancer cell, we're also eliminating the normal bone marrow. And when we eliminate the normal bone marrow, we also eliminate its capacity to make the blood-forming cells. So therefore, the patient relies on transfusion, red blood cell transfusion for survival, platelet transfusion to prevent them from bleeding, and they don't have any white blood cells, and we can't replace the white blood cells. So we have to find a source of cells, a source of marrow, to replace the blood system and the immune system. Otherwise, the patient is going to be dependent on transfusion all his life and actually will likely suffer from very important complications, particularly due to the fact that he doesn't have white cells. So how do we find stem cells in order to replace those cells that are actually killed? Well, we can go in the family and look for a brother or sister that's HLA matched, so match for the white blood cells. And by the way, this is different from red blood cell matching. So white blood cell is quite complicated. Many groups were all different between each other. However, the ability to find a donor in the family is quite restricted. Approximately 1 in 4 has somebody in his family that's compatible. So what do we do then? Well, we look for what we call an unrelated donor. So we go in the World Registry and we try to find a donor that has the same white blood cell group. So throughout the planet, there are millions of people that accepted to give their blood group in order to be able to give their stem cells when needed to patients who need a transplant. Unfortunately, even with tens of millions of donors throughout the globe, we are still not able to find a donor for everyone. And still about 30 to 40 to 45% of the patients actually don't have a donor. Fortunately, which only a few years ago, this was really problematic and we didn't have a solution, but over the last years, there has been significant development allowing us to use umbilical cord blood as a source of stem cells for many of these patients. Alternatively, it's also possible to use somebody of the family. So you use somebody that's not perfectly matched if he's only 50% matched. For example, The parents can be used as donors for their kids, or the kids for their parents. And there are issues, obviously, when you use grafts that are not perfectly matched using umbilical cord blood or haplotransplant. And that's some of the issues that we're going to discuss, which actually illustrates the activity of the immune system. Now, this is using a donor. I also want to indicate that some of you have heard or have undergone an autologous transplant, particularly in lymphoma or multiple myeloma. And so there is the possibility of doing this stem cell transplant by taking the stem cells from the patient before doing before giving the chemotherapy and radiation, freezing down those cells, treating the patient, and then thawing the cells and administering them to the patient. Unfortunately, as you can imagine, if the bone marrow is full of cancer, it's hard to take those cells. So it's only limited to those in which we can clean up the marrow to a sufficient level that we can eliminate the majority of cancer cells. Today we are going to talk only on the first possibility, which is the use of a donor, and therefore taking a clear advantage of the immune system. And what we are doing in all of these instances is actually to collect stem cells. By the way, these are usually collected by peripheral blood, so we don't have to go to the operating room anymore, or very rarely. Most of the instances, we simply administer a growth factor in the blood, subcutaneously actually, and in the blood, the stem cells rise in number. Then we collect these cells, and these are the cells that we will administer to the patient in order to regenerate red blood cell production, white blood cell production, and platelet production. So I was telling you that there is a new possibility, which is using donors that are only half matched to the patient, that is haploidentical donor. What's the issue there? Well, when we take the cells from the donor and administer them to the patient in order for him to reconstitute a blood system, what we are giving is actually cells within which there are some that have the ability to recognize the patient's own normal organs. That means the liver, the bowel, the lungs, the eyes, and the skin. And this reaction can be very severe. However, there are other cells which are able to react to organisms that we want to get rid of. For example, viruses and fungi. And the third slide, the cells that are in blue are actually leukemia cells. So actually these white blood cells do play a major role in preventing the infection and also from fighting the residual leukemia cells that the chemotherapy and or radiation may have left behind. Now what we want to do is be able to actually eliminate those cells that attack the normal tissues and actually preserve the good white blood cells that are fighting infection and fighting leukemia. Are we able to do that? Well, we've actually developed here at Maisonneuve-Rosemont Hospital in Montreal a strategy where we are taking patient cells and showing them to the donor before the transplant. So we take these cells from the peripheral blood and bring them to the lab, and in the lab expose donor cells to basically the tissue that would be seen once these cells are administered to the patient. What happens when we do that is actually that the donor cells, the cells that are able to recognize the antigen of the patient, which are not all of the cells, there is a subgroup of cells, and these cells become activated. So you see here, this is schematized in red, so the cells become activated because they see the antigen. Now, interestingly, when we expose these cells to a dye, which is called TH9402, so a rhodamine dye, these cells accumulate a dye, and in contrast to the cells that are not activated, that are not do not have the ability to actually hold down this dye. And what happens is we actually found out that what happens is that the activated cells shut down a channel which actually pumps out this drug, so the drug actually accumulates in those cells. Now you're wondering, why are we using a dye to color the cells? Well, actually what is interesting is that this dye has very special properties. When we expose the cells to light, the dye captures the energy and delivers it in a fashion that it kills the bad cells, the cells with the ability to recognize the patient, and leaves around cells with the ability to fight infection and fight leukemia, and that's what we are using in stem cell transplant. So with that being developed, we initiated what we call a phase 1 clinical trial, where we took patients that actually had failed a number of other treatments and that did not have a donor, either a world registry donor or a family donor, and we had them undergo the haplotransplant I was just talking to you about. They received intensive chemotherapy and radiation, were administered first a stem cell transplant devoid of T cells, of immune cells, and then after the transplant, we gave increasing doses of the cells I was just talking to you about. The idea is really to generate an immune system with the ability to fight infection and to fight leukemia, but prevent those cells from attacking the patient. And interestingly, here we did that without using any immune suppression at all. So we were only relying on the ability of the treatment in the lab to kill those cells. So here you have the result in what we call a survival curve. So you can see that the patients from the early cohort, so those who received small numbers of cells, and don't worry, you won't be quizzed at the end on asking you how many cells are needed. Don't worry about that. But what you should remember is that when we gave low numbers of these cells of the immune system, actually we had some interesting early results, but in the long term, many of the patients either developed infection or had relapse. But interestingly, the patients that received higher numbers of cells, which is cohort 4 to 6, actually had a survival in the area of 70%, which was extremely interesting as these patients did not have any other therapeutic option available for them. Interestingly, something we did not want to see is that if we further increase the number of cells, what we started to see was actually some graft versus host reaction. So some of the donor cells were reacting to the patient cells, and actually the outcome was not any better if we gave more cells. So actually we were able, with this study, to identify a number of cells that could be administered safely to patients with extremely good results. That phase 1 study was done in one center at Maisonneuve-Rosemont Hospital, Montreal, and it needed to be confirmed in a larger study. So we therefore conducted a phase 2 international trial in a larger patient population, and that was done both in Canada and in Europe, so throughout many centers. And what we have shown is that we were able to do this without causing significant grade 3 or 4 graft versus host disease. So graft versus host disease, or GVHD, is what I was just telling you about, which is the disease where the donor cells attack the patient. Now, this can be very severe, in which case we call it grade 3 or 4. And as you can see in the graph on the left lower section of the slide, you see that the number of patients developing acute severe GVHD was actually 0. There were a few that developed low-grade GVHD, but this was highly controllable, and patients were able to come to have this resolved. There was one patient that developed chronic severe GVHD, but the number of patients developing this was therefore very limited. Most interestingly, although all of them were actually acute leukemia patients that had to undergo a transplant, the relapse rate was very low, as you can see in the graph on the upper right panel. And clearly this was very encouraging. Actually, what we wanted to do is actually to compare that to what is the best treatment that was available, the same treatment but without the addition of cells of the immune system to fight the leukemia and fight the infection. So the curve that you see in green is actually that was used in the trial that I just described to you. And the curve in blue is the outcome of patients that were actually treated with the same therapy but without the administration of the cells. And this was done somewhat slightly earlier than doing or simultaneously in patients who could not undergo this treatment. And clearly you can see that there is very interesting difference. Now this is not a perfect comparison because it's not the same study. So it was done in two different studies. The green curve was done separately, and therefore it would be ideal if it was possible to randomize patient to one arm over another. This has not been done and is currently the topic of a study that we have just started, which is comparing this treatment actually not to the blue curve, but actually to the best treatment that patients are able to receive worldwide. I want to show you a picture because I think it's very nice to show you graphs of survival, and you know, everybody's a dot on the curve, and we lose the image or the idea of what is happening to individual patients. So actually the young guy on the left, Eric Kudelka, is actually has been one of my patients for more than 10 years now. And this individual was going around with leukemia cells that could not be eradicated. He got tons of chemotherapy, still the leukemia was left around. He didn't have anybody in his family. He didn't have a donor around the planet that could be used. So we actually used his father, which was only half matched to him in order to do this transplant. And this was done using exactly the same protocol I was just describing to you. And probably, actually, Eric is probably listening in on this webcast. So if you're there, hello, Eric. His father actually gave him the marrow under this protocol, and his donor cells were able to fight the leukemia. So this is really extremely impressive, and to me is worth 1,000 words, and I think is worth all the curse, because this leukemia could not be cured by all the chemotherapy that we could use, but using cells of a donor, his father's cells could fight the leukemia and get him back into remission, and that to me is very telling. I want to go further. We're talking allogeneic hematopoietic cell transplant, so AHCT, that's what it stands for. And I told you initially that I would be talking about MIAs, which is minor histocompatibility antigens targeted therapy. So don't be disturbed by the letters. We'll go over that and you'll understand everything. Now, what's interesting is that despite the success of transplant, this therapy is quite rudimentary, and it's toxic, and there are patients who are still dying from the procedure. So it's not a benign approach. And the idea is that we're administering immune system cells, but actually they're not selected for the ability to recognize leukemia cell. And I was showing you just now the example of how we can select for some cells, eliminate those cells that cause damage. But what would be also very interesting is, could we select those cells that have the ability to react toward cancer cell in a selective fashion? So one target that we have been using is MIA, so immune cells reacting against minor histocompatibility antigen, which are what we call non-self peptides or protein fragments. And peptides are proteins that are cut in small pieces that are present on the surface of cancer cells. Interestingly, so it's possible to target these antigens that are actually in yellow. In the top panel, what you see is a green cell, which is actually a white blood cell, which we would call a killer T cell, that has a receptor, which is called TCR, for T cell receptor, that has the ability to recognize small portion of proteins which are present on the surface of leukemia cell. And you have below the leukemia cell, which is in pink. And on the surface of the leukemia cell is the peptide that's being presented by something that looks like a hand, is actually holding up this peptide so that it can be recognized by T cells. And that is the target that we wanted to eliminate, and that provides us with the ability of the immune system to really fight leukemia and other cancers. Where do these antigens come from, like the minor histocompatibility antigens? Actually, they come from the normal machinery of our cells. Our cells are working, and they are putting on the surface small antigens that are being presented by molecules that allow the immune system to function. And it is these minor histocompatibility antigens, these portions of proteins that are expressed on the leukemia cell. So on the left is the leukemia cell, and it could be another cancer cell, versus an immune cell. And here is the T cell, so T cell subset. So among the white blood cells, there are what we call T cells, which are highly specific. And these cells are so specific that they have the ability to recognize this very small peptide on the surface of the cell. Now, don't be misled. In this schema, you see only one peptide. But actually, on the surface of the cells, there are thousands and hundreds of thousands of these molecules that can be recognized, and therefore the immune system has the ability to really kill the cancer cells. Now, my colleague Claude Perrault at the IRIC, Montreal Institute for Research in Immunology and Cancer, and also at the Maisonneuve-Rosemont Hospital, has been working on minor histocompatibility antigens all his life. And Claude was able to find a way to generate T cells that would fight these leukemia through these antigens that are expressed on the surface. He initially identified some of these proteins that were found on animal leukemia, and he was able to show in the panel on the left that he was able to cure the patients or the animals that actually had developed leukemia, while the control animals that had only the leukemia and actually didn't receive those cells had a very early mortality. And you see here that they basically all die within 40 days. Whereas the group that received the treatment anti-B6-TUM are all surviving. Now, interestingly, it's not only useful in leukemia, but it's also useful in melanoma and bringing responses in what we call solid tumors. Now, we wanted to bring that to patients, so it was possible. So Claude and a team of researchers identified a number of peptides so that the leukemia cells could be targeted. And with the help of Jean-Sébastien Delille here at Maisonneuve-Rosemont Hospital, we developed a strategy for growing up immune cells which have actually the ability to specifically target and fight leukemia cells. So what we're actually doing is we're manufacturing in the lab anti-cancer cells. And what we want to do, with the help of the funding agencies named here and lots of other people, is actually when patients with leukemia who've already undergone a stem cell transplant. So we really take the worst patients, the patients that have really tried, I would say, the most potent strategies and failed this strategy. What we want to do is go back to the donor, take the white blood cells of the donor. We want to vaccinate those cells. So we want to expose them to parts of the leukemia, grow the cells in the laboratory. So the T cell I was talking to you about earlier, well, these T cells that are able to recognize the leukemia cell, we're growing them in the lab, and then we will be injecting them into patients with leukemia so that they can fight the leukemia. So there is promise in order to advance, and I would say that strategies such as these can only be done if we do research. And I want to emphasize and thank everybody who, and you can see here the funding agencies. Leukemia Lymphoma Society is actually raising funds for helping develop research. Clearly, research is a major emphasis for developing novel strategies that are extremely promising and will come to the market. Why am I so excited about this strategy? Well, I can tell you we're doing already a clinical trial where we're taking cells from the immune system to fight another cancer, and that's what we call post-transplant lymphoma. So patients who've had a transplant or those that have a kidney transplant or solid tumors, many of them develop a lymphoma which is caused by a virus. And what we've been doing is administering patients cells that have been vaccinated against parts of the virus. So we actually take antigens from the virus and do exactly what I showed you before, expose them in the lab, expose the donor cells to these parts of viruses, grow the cells with the ability to recognize the virus, and that in a study led by Jean-Sébastien Delille, we're administering that to patients with this lymphoma. And actually the slide that you can see, the panel on the left, is a real patient that failed chemotherapy to treat this lymphoma, but had cells, and as you can see on the right, was able to clear the cancer with cells alone, without any chemotherapy. So I think this is another very convincing example of the power of the immune system to fight cancer. Now, right now, a lot of people are saying, yes, but this is really complicated, and you're absolutely right. This is done right now manually, like as you can see on the left, in areas which are highly controlled, where we want to make sure we're not contaminating the cells. And gradually, what we are working on is actually to develop robots and use actually bioreactors and other strategies to make this an industrialized fashion so that this is more amenable to treating large number of patients. Now, I want to take a few minutes to go over another strategy of the immune system to fight cancer that's really, really very exciting. And that's modifying cells in a slightly different fashion. This is using what we call chimeric antigen receptor technology, or CAR technology. And this is one you can drive, but not in the usual fashion. What is happening, and you can see on the left, is actually, again, a white blood cell, which is actually for this picture colored in orange so that you can see it, but that is binding to a cancer cell in pink or purple. And you can see that it's not only hugging the cell, but if you waited, you would see that the cell in purple actually dies. So what happens? What did we do differently? Well, the cells are actually infected with what we call a vector, where we introduce part of the DNA of a new molecule. And the cell makes a new molecule that goes on the surface of the white blood cells and will now be able to target antigens that are expressed by the cancer. So we are making the donor cell, or making the cell, actually, which comes from the patient, able to react specifically to antigens present on the surface of its cancer. What are we targeting? Right now, we're targeting what we call CD19, cluster designation 19. So you don't have to remember that. But what you have to remember is that this molecule or this antigen is present on lots of cancer cells, and you can see them listed or abbreviated: CLL, chronic lymphocytic lymphoma, leukemia, B, acute leukemia, diffuse large B-cell lymphoma, follicular lymphoma, mantle cell lymphoma. And why is that? Well, this is because this antigen is present on all the cells of what we call the B-cell lineage. So this is part of our hematopoietic system, so the blood system. So on the left, you have a stem cell, and the stem cell gives rise to lots of cells, among which there are some B cells, and there's like pro-B, pre-B, B cell, etc. And these cells are normally producing antibodies to prevent us from having deadly complications of infection. But in this strategy, what we are using is actually killing those B cells that can be replaced, by the way, killing the B cells that are the cancer cell. And this antigen is present throughout these different stages of maturation. So here you see a B cell growing from kindergarten to high school, to graduate school, to university, and then to the working place, and actually being on the workforce. And you can see that all of them are all expressing CD19 and can all be killed by this strategy. So how does it work? Well, it has, and you can just look on the right, it has a Y structure, which is orangey, a dark orange, which is the antigen binding domain. So we plug onto the cell a receptor that has the ability to attach to the cancer cell. But that by itself is not sufficient, and researchers have been working on this for many, many years, and just binding is not sufficient. What it has to do is to activate the immune system and really send a signal that this is working. And on the right, and you can see below, the two structure for 1BB and CD3 zeta, that's an example, but they're what we call signaling domain. And what they do is when the upper portion is binding to the cancer cell, it sends a signal to the C cell to kill its target. And does it generate interesting results? Where here you see the survival in a pediatric population, 80% of the patients surviving post-injection of these cells in patients who had leukemia, and the patients are in complete remission. So this strategy is not only very promising, but it has actually been accepted in the United States and is currently under review in Canada for implementation for usage in patients. And this is generating a lot of enthusiasm throughout the planet on actually a novel way of targeting cancer. And as I was telling you earlier, this strategy can be used to target a lot of different targets, and this is extremely interesting for us oncologists who are treating patients with blood cancer. I also want to tell you, as you can see, there are lots of advances in the area of immune therapy. And one I want to talk to you about is what we call checkpoint blockade. And by now you're all immunologists, and you now understand that the T cell is doing a great job, and the T cell below is actually binding to the tumor cell, and you all know by now how it works. But what I didn't tell you is that on the left there are signals that are generated by other molecules, one of which is PD-1, which is actually binding here to PD-L1 as an example. And what this binding says is it tells the T cell, well, don't be too active. And the reason is we want to limit the ability of our immune system so that it doesn't go overboard. So what it is actually it's a foot on the brake of the immune system. But there are now ways of removing the foot on the brake in actually allowing these cells to be more active. This is used efficiently in treatment of melanoma and advanced lung cancer, and you can see in both curves that there are improved. The curves using this strategy are those in blue, and there are improvements over other strategies, which is extremely promising and very interesting for patients. And that is currently being used in the treatment of patients with cancer. I was telling you about PD-1. You can see here there are many other molecules that are actually preventing the T cells from being at its best against cancer that can be targeted. And on the left, there are a number of activating receptors that could also be stimulated so that we increase the ability of the immune system. Interestingly, one of our researchers here at the hospital has developed, Christopher Rudd, has developed a new molecule, which is XL334, a new system, actually, a whole new system that is based on the strategy that I was just telling you. And on the right, you can see that when we administer cancer to animals, and these are cancer cells in the lung that you can see in the black, the B16 no treatment, you can see that if we add novel agents, such as the one inhibiting GSK3 or anti-PD-1, we see a decrease in the number of cancer cells, but when we're combining with both, we actually see very interesting results. So these are the types of findings that are really taking us a step further. I also want to say that it's also possible to expand stem cells and produce stem cells that are probably more active against the cancer, and you already know how transplant works, so I'll go very quickly. But actually, Guy Sauvageau and Sandra Cohen are actually putting into patients a strategy where sometimes we don't have enough stem cells. So what is being done is actually to take stem cells right now from umbilical cord blood, exposing them to a molecule called UM171, growing the cells in the laboratory so that we have lots of stem cells, and then can take these cells to grow white blood cells. So cells are being used in transplant right now to help patients go through a stem cell transplant, and results are extremely interesting. In order to achieve those strategies, we are able to do that because we have a dedicated facility, and there are a number of them across the country. The largest one is in Montreal, and it's a whole building totally dedicated to the production of stem cells. So this building, which is at Maisonneuve, Rosemont Hospital, is actually a 4-story building where there's a floor where people are going around gowned like what you see here in order to prevent transmission of infections. For the production of cells that I was just talking to you about, there's an area for freezing the cells and for verifying the quality of the cells, and actually 2 floors of hair handling system and sophisticated electromechanic apparatus to make sure that this is working. We've also developed a Center for Commercialization of Cancer Immunotherapy, so not commercialization in terms of advertising, but how can we bring interesting developments that are at the bench side and bring them so that patients can benefit into clinical trials and further development. We have a vision of making Canada a catalyst for cancer immunotherapy, and we really want to accelerate the discovery and commercialization to cancer immunotherapy approaches, and that's called C3I. And it's what we call a one-stop shop with a biomarker unit, a cell manufacturing unit, a clinical research unit, and an innovation commercialization unit. Also, I would like to invite you to go and visit our CellCan website that you just heard about initially, which is actually a site where you can get news of the use of stem cells, not only for fighting cancer, but actually for lots of other usage. You have the website is www.cellcan.com, or if you fail to remember that, just type in "I love stem cells," and it will take you to the right website, and you can have news on stem cells and links with our partners. We also have a number of booklets for both health professional and the general public on various aspects, stem cells, medical tourism, how to take these cells into patients. We have an app which is called Regi, which you can download from the App Store. So I would invite you to just type in Regi, and you'll be able to have an app that tells you more about stem cells and their usage for the treatment of various disorders, and where we are currently in advancing this towards the clinic. And there's actually a role for protecting Canadians from unproven therapies, as there are lots of people that are claiming activity either from the immune system or from stem cells, and I would invite a lot of caution on these various therapies. Clearly, you're putting your safety at risk when you are accepting those treatments, so please tell those who are envisioning treating to get informed and really to be wary of the fact that stem cells could be dangerous if not used in the appropriate fashion. So I would invite you to stay informed. If you have questions, you can go to the various websites, www.cellcan.com. Soon we will give you that of the Leukemia and Lymphoma Society. You're welcome to contact us at Cellcan. There's a help desk, and also to follow us on Twitter. And I would like to end on acknowledging a number of people that are involved, and you can see really it's an international group, pan-Canadian and international, both for treating patients, for manufacturing, for laboratory, but also behind the scenes, lots of people that are really helping, administrators, developers, builders, administrators, businessmen, donors, and all of you. This is really crucial, and on the right you can see our funding agencies, and we want to thank all of them for really their great contribution. So at this point, I'm ready to take questions, and I would give the podium to my host.
Well, thank you very much, Dr. Waugh, for showing us some of the advancements in research. I particularly want to thank you for putting faces on research. Eric Puzelka, I also saw a picture of William Brock there. You know, it really sheds light on the importance of people that are benefiting live from this research happening right here at Hôpital Maisonneuve-Rosemont. So thank you very much for that great presentation. The Leukemia and Lymphoma Society will continue to do its part to continue to speed up the discoveries of better treatment options. And now, I guess we'll open up to the floor after this great presentation. I'm sure there's a lot of questions out there, so we'll take maybe a few seconds to review some of the questions to make sure that we are addressing the recurring questions. I will read the question, and Dr. Waugh will then answer it. So the first question we have here is immunotherapy being done in Toronto?
Absolutely, no hesitation. I think I was giving you examples of projects that actually been developed at HMR, some of which are not developed at HMR, but at different places around the planet. What I would like to say is, one, a lot of the programs that I just presented to you are actually also performed in Toronto, and they're done collaboratively with the teams there. And also, I would like to underline the fact that there are researchers at Toronto that have also found different approaches to target leukemia. So we're all leaguing together to try to find ways to fight cancer, and clearly there's no one way to do this, but there are many approaches that are being used, and we need actually to develop novel therapies if we are to fight and actually to cure cancer, not only in Toronto, but in other places across Canada, Vancouver, Edmonton, Calgary, Ottawa, Hamilton, Maritimes. So I would say throughout the planet, this is not restricted. There are researchers across the country and across the globe that are working very actively on these various options.
So the next question here is, I think, a question we hear often in news about CAR T therapy, and where the question comes is as follows: how close is CAR T to be available to adults to treat lymphoma in Canada?
And that's a very interesting question. I wish I knew, but what I can tell you is it's already approved in the United States, and it has been applied for in Canada. So we are confident that it will be approved soon. When, I do not know. Clearly, regulatory agencies have to go through all the documentation, make sure everything is in line, and it will clearly be possible to treat Canadian patients with this technology in the near future. I wish I could answer the question more precisely, but I think what the good news is, it's going to come soon.
I'll ask a general question here. Is immunotherapy the future of cancer treatment?
That's a very astute question. Actually, this immunotherapy is what has generated the most, I would say, buzz in the last couple of years, but it's not only a buzz or a transient, it's effect. It really is using something that has been around. We have our immune system, we have the ability to fight cancer, and now we are finding ways of really taking maximum advantage of the immune system to fight the cancer. And that is clearly a significant advance. It's not there to go away. Clearly, already, for example, the checkpoint inhibitors are in clinic, and they are being used for treating a large number of patients with various cancers with very interesting results. And it is coming in many diseases. We're talking CAR T cell, we're talking specific cells in the area of stem cell transplant. Like, all of these are developing, and clearly this is around to stay. Maybe I want to give a word of caution. Like, we're emphasizing the success of this strategy, but I want to emphasize that still there are patients who fail. Still there are patients who are not doing as we wanted. There are patients who have toxicity associated with these various therapies. So we want to develop strategies that are highly efficient but have low toxicity. That's not easy to develop. I think we're getting closer to that, and clearly with these novel immune therapies, we're able to decrease the toxicity, decrease the side effect. Still, it's not 100%, and that's what we're aiming for, but there is still room for improvement.
So, a question we have here: what role do drugs like daratumumab play?
Yeah, well, there are many drugs that I didn't speak about that are highly efficient in various cancer. Clearly, there are antibodies that are highly efficient. I want to specify this one, but there are many antibodies that are being used in various diseases: multiple myeloma, lymphoma, leukemia. So I don't want to be specific on the antigen and the antibody that can be targeted. I can tell you that we have benefited already from immune therapies in the use of monoclonal antibodies. This has been around. This is actually evolves from the immune system. The antibodies are produced by our B cells, by our immune cells, and the antibodies that are being produced are actually a modified form of the antibody to target the cancer cells. So this is all the strategies that we've been talking about are not necessarily to replace, although they could, but in many instances, or in most instances, is going to be an add-on, because clearly cancer is a tough nut to crack, and if we want to really cure, that's our objective. We need to put all our strength to do that, and we need to put all together all the different strategies So there's surgery, there's chemotherapy, radiation therapy, antibodies, cell therapy. These should all be factored in, checkpoint inhibitors, and others. And there are many that we didn't talk about, but they are useful.
We have a little more of a specific question here and an example. So the question is, I am 63 and have MDS. I have two brothers that are not full match. With the advancements that are being made, are you saying that there's still possible stem cell donors if I need a transplant?
I think that there is the possibility of doing transplant in the form that we were talking about in many centers. I would say up to age 65 and sometimes older. I think this has to be discussed with your physician, with your oncologist. Clearly, there are various options that are available. This may not be the best for you, or it may be. I do not know. It's impossible to comment with limited information, and sometimes even these things have to be discussed in group with the whole chart, knowing exactly what are the details of the condition. So I would say please refer to your oncologist, have the person, your oncologist, look into the various possibilities. I'm sure they are, and for knowing all the oncologists I know are really taking good care of their patients, and they are taking this at heart, and that's why we're doing this. So they are the best ones to answer your question, and if they're not doing that at their center, they will be able to refer you to a center that will be able to answer the question and see what type of treatment they could be offering.
The next question here, I think I will answer part of it. It is a question about clinical trials. It is, how can I find out whether clinical trials in CAR-T therapy or checkpoint blockade therapy? So I would like to remind the people online that are not aware of our clinical trial number. We do have a 1-800 number available that anybody can call to get information on clinical trials. If you want to have more information about that, you can definitely visit our website. You can contact your local patient education service manager in your region. He will be able to give you all that information by visiting our website, lscanada.org. And if you do have any questions about clinical trials that are available, we do have specialists that are in association with the LLS US that will answer all clinical trial questions about the US and also for Canada. So feel free to get those answers on our website or by contacting us directly. So the next question we have here, is there any treatments available to slow down the growth of white cells without going through chemotherapy?
I think we've been discussing some of these. There are clearly, as I was mentioning earlier, a number of other treatments that are developed. So I would like to emphasize that we're talking about a anti-cancer modality, but there are various strategies that are attacking cancer cells. There are molecules that go into the signaling pathways, some of which are affecting the growth of the cancer cells. Some are not necessarily curing patients, but are actually helping patients survive and live longer. So it's not always possible to achieve cure, but there are many strategies that are out there, and many more that are being developed. What I would like to leave all of you with is a message of hope. I think researchers are working very hard. Clinicians are working very hard to put these to test so that we are offering our patients the best treatment options. Clearly, we have not won the fight against cancer. We've done major advances. We have taken this a long way with immune therapies, we've opened up a new area, and it's brought a lot of enthusiasm. I can tell you the curves have shifted in many cancer in a way that we haven't been seeing in the last 20 years. This is a major advance. This is the message that we wanted to convey to you, that one, there is hope, two, there are significant advances with immune therapies, and clearly we should be happy with what we have achieved, but we should not be satisfied. We should continue working to get to even higher levels of successes, and as you can see, this can be achieved.
Thank you very much once again, Dr. Roy, for your presentation. Thank you very much for your message of hope. I also would like to thank Paladin Artists for the educational support that made this webcast possible. Earlier this afternoon, we sent you an evaluation form, so please fill it out to make us better understand your needs and services so that we can bring you other webcasts such as these that'll be able to answer the questions you have about our services and our research programs. I also invite you to visit our website at lls.org, and I will remind you that the webcast will be available online, and you will all be sent an email once it is available. Thank you once again, Dr. Roy. Thank you, everybody, for joining, and I wish you all a very good weekend.